Bright Duty. Every student matters.
So now the next topic is in detail study about the plant tissue. So this flow diagram will actually tell you, will give you a nutshell about how plant tissues are divided. Plant tissues are divided into two main categories, the meristematic tissues and the permanent tissues. Meristematic tissues are those which can perform cell division. They are metabolically active. They are continuously growing and dividing. So they help in the growth of the plant. While on the other hand, permanent tissues, they do not cause cell division. They have taken up a certain permanent shape and size and function. Meristematic are further divided into apical, intercalary and lateral. Alright, depending upon how they are positioned. We'll study in detail about them. On the other hand, permanent are divided into simple and complex. Simple are further divided into three parts, parenchyma, collenchyma, sclerenchyma, and complex are of two types, xylem and phloem. So we need to study in detail about all these types of plant tissues and how do they differ from each other. So let's do that. First is the meristematic tissue. As I told you, the growth of the plant is at a specific region, which is because of cell division of specialized cells. These cells they form the group which is called meristematic tissues. So what are the main characteristics? Cells are dividing continuously. All the cells are similar in structure. They are compactly arranged. They contain dense cytoplasm and a prominent nucleus. There are no vacuoles because they are metabolically very active. If you remember in the last chapter cell, I told you the function of the vacuole is to store food. Like whatever excess food or nutrients they have, they are stored in the vacuoles. But in meristematic tissue, since they are growing, they are metabolically very active. So that is why they do not have excess nutrients. Whatever nutrients are available, they utilize it for the growth and cell division. That is why vacuoles are absent in meristematic tissues. They are metabolically highly active. They have very thin wall made up of cellulose. They are found in the growing regions of the plant. The functions are, firstly, they are the parent tissue. Some of this meristematic tissue, they later on take up a permanent shape and size and they become the permanent tissue. So they act as a parent tissue for the permanent tissue. Second, they help in the growth of the plant, whether it is increase in the size of the fruit or increase in the height of stem and roots. That is all because of meristematic tissue. Then the next function is the healing feature. If there is any injury or the breakage of any part of the plant, then that part is healed by the cell division of the meristematic tissues. These are the main characteristics of the meristematic tissues. Look at this diagram. If you can see, if you remember this diagram, you can clearly state the characteristics. Cells are compactly arranged. There is no intercellular space. They have a prominent nucleus, very dense cytoplasm, no vacuole. All right. So these are this shape will exactly give you idea what are meristematic tissue, about their structure. Now, before we understand the types of meristematic tissue, you can perform an activity at your home. Take two jars, jar 1 and jar 2, and put two onions with small roots, such that these roots they are dipped in water. You have to fill the jars with water. Now observe the growth of these roots for 2 to 3 days. You will notice that these roots will start growing. They will increase in length because they are getting water and which is helping them to grow. Now try to understand. This growth is because of the meristematic tissue which is present at the tips of the roots. Those tissues, they will divide. The cell division will cause elongation as well as more number of cells, because of which the length of the roots will increase. Now after 3 to 4 days, what you can do, you cut the tips of the roots in jar 1 and then again dip it. Okay, cut all the tips. Now then observe it again for 3 to 4 days. You will notice that in jar 1, in which you have cut the tips of the roots will not grow any further. But in jar 2, the length will keep on growing. Why this happens? Because the tips of the root contain the meristematic tissue, which was helping the root to increase their size. Now, This helps us to show, this activity helps us to show the role of meristematic tissue. Now, meristematic tissues are further divided into three categories depending upon where they are located. First is apical meristem, intercalary meristem and lateral meristem. Apical meristem is actually present in the apex, that is tips of the roots and shoots. Intercalary, as you can see, they are present at the internodes. Okay, nodes are the places in the stem from which the branches or the twigs arise. So the gap between two nodes is called the internodes. These intercalary meristem, they are present in the internodes, and lateral meristem are present in the diameter of the older stems and the roots. They are that is why they are called lateral meristem. So according to their position, they are divided into three categories. So let's understand their function. First is apical meristem. Apical meristem, as you just saw, they are situated at the growing tips of stem and roots, and they cause elongation of root and stem. As these cells will divide, and the height of the plant will increase. Second is the lateral meristem. They are present beneath the bark, which is called cambium, okay, just beneath the bark, and they are present in the diameter or girth of the stem. So as they divide, the diameter or the girth of the stem and the roots increases. So they increase the diameter and girth of the stem, roots and older branches. Third is the intercalary stem. It is present at the base of the leaves and internodes on the twigs. They cause increase in the length of leaves as well as the internodes, that is the gap between the two nodes. So all these types of meristems, they are actually the classification is dependent only upon the location. Otherwise their function is same, that they cause cell division and the growth.